Hi, my name is Laura Mateo.
And I'm Anna Jimenez.
And welcome to the Science Corner. This lab will be about DNA extraction.
It's an assignment for Ms. Bulallen's AP Biology class in Jacqueline Kennedy Onassis High School. Now, it is our objective to
Extract DNA from a strawberry,
which is large enough to see with the naked eye. So the materials that you're going to need for this lab is a half a cup of water,
rubbing alcohol,
plastic cups,
detergent,
we're using Dawn,
3 plastic baggies,
extract, we have salt, and of course strawberries. But these specific strawberries have been frozen for 24 hours and thawed for another, so they're extra mushy. We also need a coffee filter.
kind of like this.
And a teaspoon. And of course, safety. Although we're not working with any hazardous chemicals or intricate tools, it's always best to follow the lab safety. So we have our stylish lab coats
And our gloves.
gloves. The first step of the lab is to see if there are any green leaves on your strawberries. If there are any,
Please
take
touch.
them.
Yeah.
Okay. So the first thing that you're going to do is take a couple of your strawberries. We're going to be using 3 for this lab.
And put them into a plastic bag.
Make sure that there is no air. So let's just open this up a little bit.
And release the air by pressing down.
And then you're going to gently start smashing them for around 2 minutes. If you can see, just like that. So while Lori does that, I will go over how to do your DNA extraction liquid. So the first thing that you're going to need is 2 teaspoons of detergent. Like I said before, we're going to be using Dawn soap. You're going to need the half a cup of water. And lastly, 2 teaspoons of salt. You actually need a lot of these salt packets. Last one should be enough. And then you're going to gently mix, avoiding any soap bubbles. Okay, so this is done. Now we check with the strawberries. Now with the strawberries, just a little bit more. Doing this will start breaking open the cells in the strawberries, therefore releasing the DNA. And this extraction liquid, once we place it inside, will further open up the cells, so that once we pour it into the cup, which is all mixed up, you'll begin to see the DNA.
The whole point is to open the pores.
So
this is how it should look.
the next thing you're going to do is to pour your DNA extraction liquid into here. So here we're only going to pour 2 tablespoons.
1, 2.
Once again, we're going to zip up our bag,
Releasing the air, of course.
and we're going to mix for another minute. While you're doing so, make sure there no bubbles are forming, and this will further break open the cells and release the DNA. So do this for a minute.
And make sure you're flattening.
all the
The next thing that you want to get ready is your coffee filter. There's two here. So you're going to place the coffee filter and once the minute's up, make sure there's no bubbles. We're going to place the pour and the DNA into our filter. So if you may, Lauren. Let me just move this so.
Make sure you get all of the strawberry out.
If you see that nothing's falling, you can simply just close it up and squeeze gently.
To extract the liquid.
Just be really patient because if not, the back can break or the filter. I think that's all we can get out, or maybe a little more. That's all. Next, you're going to want to pour an equal amount of rubbing alcohol into the So since it's around here, we're just going to pour half. Around there. Now, all we have to do is wait. All we have to wait and see until a cloudy layer starts forming. And if you can zoom in,
Approximately 3 layers.
you'll start seeing right here is a lighter state. See here, there's darkness. Here, there's a really cloudy layer, and here's the dark part. Now, if we just wait a couple of seconds, we'll actually be able to pick it up.
And see the different layers.
So, as you can see, the alcohol that was added was actually able to isolate the DNA from the rest of the cells in the bottom layer of the strawberry. So if we just go ahead, see it's not there yet.
To wait a couple of seconds.
And now you can use a wooden stick to pick up your DNA.
And extract it.
Your DNA, you can actually save it in a little plastic baggie, just like this. For the post-lab questions. So Lori, number 1. Why did you need to mash the strawberry? What happened?
Basically, you mash it so that it produces enzymes that will break down the cell wall. Did the DNA extraction buffer/lysis solution have?
Well, with the enzyme and the coupled salt, it both has the ability to break open the cell walls of the nucleus, which will then release the DNA. If we add soap to the strawberry, what would it do to it?
Well, adding the soap actually opens up the nucleus of the strawberry, which will then release the DNA. What purpose is there for filtering the strawberry buffer mixture?
To isolate the juice, will therefore release the DNA.
Good.
Number 5. Tell me about the ethanol rubbing alcohol. What observations did you make?
When we added the alcohol, it clumped the DNA together and it made it visible, so it made a layer. The interface where the filtrate in the cup and ethanol meet. What happened?
Well, in that layer, it was the regular juices that we extracted. Then there was a cloudy layer, and then there was the rubbing alcohol layer. And that cloudy part was actually the DNA.
Okay, so
Which
just
answers
answer
number
the
7. Number 8. Why would we want to extract DNA? Have you ever heard of scientists using DNA, and what did they use it for?
Yes. Scientists use DNA basically for forensics, which is finding out the criminal, and for paternity tests. And they extract the DNA to figure out the genetic composition and compare it to other organisms. Do you think the DNA extracted from a strawberry is pure enough to use in other biotechnology applications, such as PCR or genetic engineering?
No, it is not pure. Although we did extract a large piece of it, it still needs to be examined under a microscope before it can be used for other genetic testings or PCR. Now, for the last question, is summarize the experiment. Include what each material and step of the procedure did to help extract the DNA from the fruit. So the first thing were the strawberries. We use the strawberries because they have really large genomes that we can easily smash in a little plastic baggie.
And then we used a filter basically to filter out the DNA.
Yes. And then for our DNA extraction liquid, we use 2 teaspoons of salt, 2 teaspoons of detergent, and half a cup of water. And this extraction liquid will help further open up the pores of the strawberry once you place it into the Ziploc bag. And then after all that, after all it's mixed and we place it into the filter and we squeeze like we did in the beginning, we'll see that DNA form.
Yeah,
And then finally, the last step, when placing the ethanol, that will help clump up the DNA, which creates that second layer in between where we can then extract it and take it out.
And then you have DNA to save.
Yeah.
And remember, when life gives you strawberries.
Extract DNA.
And this has been
The Science Corner.